we can begin with our first talk, which will be given by Dr. Stephen Hyman, who is the director of the Stanley Center for Psychiatric Research at the Broad Institute. And they are using a variety of methods to study psychiatric disorders in terms of their genetic contributions and other biological mechanisms. So thank you. I hope that's a decent
paragraph. More than enough. Thank you very much, Ruth, and thank you, Paul. I really enjoyed yesterday, and it's a shame that NHGRI has decided that all the ELSI problems are solved, because I think there's probably, as you'll see from my talk, there are plenty of things that will raise concern. But let me just begin. I assume everybody in this group has seen a Manhattan plot, but this has become sort of iconic of the age of modern medical genetics, but especially psychiatric genetics, because we've perforce begun with the study of common variants by this methodology. So this is a Manhattan plot, and at the bottom are just the chromosomes arrayed, 1, 2, 3, so forth. And the y-axis is the statistical, the probability that the association is true, and this mythical red line is the equivalent of a probability of less than 0.05, corrected for 1 million comparisons. The most recent schizophrenia GWAS, and it's the one I'm going to show you and talk about most, because this is the psychiatric disorder that is farthest along, partly because historically a decade ago, people in the field decided that they should try to succeed with one disease instead of diluting efforts. But it also turns out that because people with schizophrenia are often so ill, they are under care, and it has been relatively more straightforward to ascertain people with schizophrenia than many other psychiatric disorders. But at any rate, there are now 287 genome-wide significant loci, and this data freeze has about 76,000 cases. At least our goal is to get to about 250,000, at which point, while there are no guarantees, we think we will saturate the biological information that we can cost effectively get. It's important, however, that as costs of DNA sequencing have come down, it has been possible to find ultra-rare, highly penetrant variants for schizophrenia, as have already been found for neurodevelopmental disorders, many of which also you heard about yesterday, actually, also associated with ASDs, some with epilepsy. So these, the ascertainment of GWAS has typically used microarrays. It's very inexpensive, and this is why it proceeded first. And it could use microarrays because these are common variants in the population with an allele frequency typically of greater than 1%, whereas penetrant variants are ultra rare. You heard from Wendy yesterday that they're essentially all de novo, and these are either de novo or near de novo in populations, because they do having a severe behavioral disorder before beginning in your teen years decreases reproductive fitness and the likelihood of passing these alleles on. So these take a lot of cases and a lot of sequencing, because you would find precisely 0 on a population-wide microarray. Right now, just published a month ago, is the first instantiation of this. What's called a schema consortium, but it's finding ultra-rare protein truncating variants in schizophrenia. 10 are now exome-wide significant. There's another data freeze that should be complete by the end of the year, And it looks like these yellow dots below the significance line, I mean, some will go away with Winner's curse, but it looks like there'll be 30 to 40 of these, and it won't exhaust the number. What's interesting about these is that they confer a very high odds ratio between, say, 2.5-fold in risk for schizophrenia for HERC1 and a 40-fold increase in risk for some of these. Of course, again, some of that effect size may decline, but it may also increase with larger numbers. Just to compare, and this is going to be important for prediction, obviously, common variants that you would get from a Manhattan plot. This is just a localized bit of a Manhattan plot. So a few genes have both common variants that you find in a GWAS and rare variants that you can only find by sequencing. One of them encodes the NR2A NMDA receptor subunit, which is really fascinating because that is a molecular target of ketamine. But the common variant that increases risk of schizophrenia has an odds ratio of 1.07, whereas a loss of function in this gene creates an odds ratio of almost 25. These are extremely, these protein truncating variants are extremely useful to neurobiologists as experimental tools because you can knock out one copy in a mouse or in a non-human primate, or make or patient iPS cell lines, or use CRISPR to create these variants against an isogenic background. So these things are beloved of biologists, but we don't quite yet know what they mean. This is a summary of the allele frequency in the general population for schizophrenia. So at this end, where the allele frequency is high, here's an allele frequency for the minor allele, often the risk allele of 0.5%, whereas these are very, very rare. And the most penetrant are the rarest, unsurprisingly. And all of the GWAS results are here at this end. And we're just getting a picture now of the variety of genetic predictors. So that sounds like a great story, but here is the first important problem that comes up, which is you probably know that genetic risk is shared across psychiatric disorders. Schizophrenia and bipolar disorder share 65 to 70% of their common variant risk, the kind you would detect on GWAS. But even highly penetrant alleles, notably, we've known for a long time about copy number variants, like the most common of which is a microdeletion that takes out there are two versions of either 38 or 44 genes on chromosome 22, but the very same copy number variant can give you either, in about 20% of cases, autism onset in infancy, schizophrenia onset in teen years that has been studied and even has a second cognitive decline during teen years, and or comorbid ADHD and a lower IQ than the biparental mean would predict. So these are really pleiotropic. They have multiple effects. They can give you multiple phenotypes. And not all, but many of these new protein truncating variants can also give multiple phenotypes. Not shown here, but very excitingly, we have the first one for bipolar disorder, a gene called ACAP11, which is an anchoring protein for protein kinase A, and also, interestingly, for a putative target of lithium, GSK3 beta. But that's a risk either for bipolar disorder or for schizophrenia. So it's not just that there's genetic sharing among common variants, but there are pleiotropic effects of these more penetrant genes. And the current best scientific insights are that it's the interaction of these more penetrant genes, for those unfortunate enough to have them, with the polygenic background that might elevate what would have been a sub-threshold risk into one or another phenotype. But there's a long way to go to understand this. So for common variants, you perhaps have seen these interactions already. But again, if you're thinking about predicting from GWAS results or polygenic scores, this complexifies the task. And so here, bipolar disorder and schizophrenia share, a large fraction of their common variants, and now they share a rare variant, ACAP11. But this is now published 4 years ago. It's just a summary of these interactions of shared risk. OCD and anorexia nervosa is significant, ADHD and major depression. This would not surprise any clinicians, by the way, shares genetic risk. Okay, so that level of complexity we're starting to get used to, but here is something that every clinician, at least when I was still seeing patients, was very troubling, and which has been pushed under the rug in a lot of genetic and biological research. So we have a cohort of 10,000 people with psychosis spectrum disorder in Finland. It's called the SUPER study, and the principal investigators are Arno Pelotti and Mark Daly. And this is somebody who has one of these penetrant mutations. It's one that confers a lower odds ratio, HERC1, but you could see this with people who have no such penetrant mutations. And what you see is that this, in teen years, this first early teen years, this proband had a diagnosis of adjustment disorder and then major depressive disorder, and then he was starting to use alcohol and substance use for any reason we can figure out. Somebody threw in a diagnosis of conduct disorder. By the way, this cohort, like all things in Finland, is linked to the medical record and the pharmacy records and cognitive testing, and so these are as reasonable as these diagnoses can get. At 24, he was diagnosed with bipolar disorder, and then at 32 with schizophrenia. And again, I've seen many patients like this. It's interesting, if you have in the DSM world both major depression and one or more anxiety disorders, you get multiple comorbid diagnoses. If you have, and we don't know that this person had shared symptoms at the same time, but if you have the same situation with bipolar disorder and schizophrenia, you don't get a comorbid diagnosis necessarily, although you may have them. You get a third made-up diagnosis called schizoaffective disorder. The problem if you're thinking about prediction, and this just absolutely demands on our part a kind of clear-eyed epistemic humility, is that this person could have been ascertained into an imaging study, a clinical trial, or a genetic study at the age of 24 and been classified as bipolar. And if you did it 8 years later, he'd be classified as schizophrenia. So the truth is, essentializing, as we heard yesterday, is probably less of a problem around genetics. It is a greater problem around the DSM and our classifications, which act as if there are these diagnostic silos, and have been therefore undermining psychiatric research for a long time. Now, we can take GWAS results, whatever phenotype you would call this is schizophrenia, but again, 65 to 70% of these variants would be shared with bipolar, a smaller percentage with MDD, and so forth. And we have to recognize that these predictions are probabilistic. They're limited by the amount of the heritability of schizophrenia that is attributable to what you would find in a GWAS, which is common variants. So let's assume that the heritability of schizophrenia is 80%. Please don't hold me to these numbers. And let's say that between 25% and 40% are due to common variants. So maybe the maximum probability that you could get from something derived from a GWAS would be 20% of 80%. Which is, by the way, useful, but is not a certain predictor. The other thing is we should be aware that this GWAS, most of the schizophrenia cases, in fact, almost all of psychiatric genetics, right now is not phenotyped well, as in the Finland study, but actually all we know about most of these cases is a DSM diagnosis, not even the age at which they got it, their biological sex, and typically their country of origin, because that tells you where the cohort was from. So it's really pretty, very shallow phenotyping. At any rate, we can sum all of the risk alleles in a population and correct each one in that sum for its effect size. So generate a weighted sum, which gives you a polygenic score. And for an individual, then you can use as the training data set, to use AI lingo, the largest GWAS that's appropriate for that person's genetic background, and we'll come back to that. And then the target data set is that individual's genotype. And then you can percentile them against their relevant population, schizophrenia risk or depression risk or any phenotypic GWAS results. And invariably, cases and controls overlap. And there are people even in the, who might be in the 7th or 8th decile of risk who don't develop the condition. Partly it's because the genes at this level work stochastically, and partly because there are lots of things we just don't understand. And similarly, there are people at relatively lower apparent risk for schizophrenia, and this is a cartoon, obviously, but it also captures the truth. Who despite being maybe in the 5th decile, still get this diagnosis. Now, it is much easier to phenotype for coronary artery disease. And so large studies have been done. And what you can see, this is percentile against the population now in percents, not deciles, polygenic score, and the prevalence of coronary artery disease. And you see this really pretty impressive curve. And then you can see here in the right tail, and this was work from Sekar Katharison at Mass General, who has gone off to found a cardiac gene therapy company. But there's a 5-fold increase in risk, which would be pretty substantial even for a rare penetrant variant. So this is starting to get action in the clinic. It's not quite there, but it's pretty clear if you have a high polygenic risk score, even correcting for smoking behavior, blood pressure, and serum lipids, you have a much higher risk. And what's actionable is that your cardiologist or even your PCP one day would dose you on statins or maybe a PCSK9 inhibitor and drive your LDL lipids way down below where normally we would have as a target. And that presumably would make this an actionable way to predict risk. Now, we also have, this is already old data, but in several Scandinavian cohorts, you can see the way polygenic scores associate with changing odds ratios in schizophrenia. It's just that at this point, this is not actionable. Polygenic scores can be applied to people. They can also be applied to cell types or brain regions. I won't take much time on this, but the schizophrenia polygenic score, even 4 or 5 years ago, the work is much better today, predicts cerebral cortex and it's both excitatory and inhibitory neurons. So what do we use them for? Well, population stratification in epidemiology. Again, these are probabilistic and partial, but if you're an epidemiologist, it's good to have any ability to stratify people if you're examining environmental risk factors. And in fact, genetics should become a really important tool for environmentally focused epidemiologists. Also for human studies, again, if you do a clinical trial and there's a subgroup that has no response at all to a treatment despite their DSM diagnosis, understanding that these diagnoses pick out highly heterogeneous and unnatural groups, A polygenic score could at least tell you that maybe this person wasn't even in the ballpark. But again, the utility you have to, is far from perfect. I've already mentioned nearing clinical utility in cardiology, and then it can be used again to map, figure out which neurons are more involved in risk of a particular disorder. Now, I've kept stressing the largest GWAS in your relevant population for the calculation of a polygenic score related to an individual. Alicia Martin has been one of the global leaders in pointing out that the diversity problem in human genetics has implications that can actually exacerbate healthcare inequalities or disparities, because just by genetic drift, almost certainly not by evolutionary selection, just by genetic drift over time, people who live apart may develop different places in their chromosomes where crossing over occurs, and therefore different patterns of linkage disequilibrium and different frequencies of particular alleles. And as a result, and this is for across, this is a sum of many disorders. If you do your GWAS in Europeans, and about 90% of human medical genetics is still done in European cohorts, it performs, polygenic scores are just about 60% as predictive in indigenous American populations, and just perform horrifically in African Americans. So you could go into a clinic today in New York and want to apply the polygenic score for risk of heart attack, but it would only really be useful for those patients sitting in your waiting room of European inheritance. And certainly we've got to do better than that. I'm almost, well, I guess we started at 10 after, so I shouldn't panic. There are, of course, as you probably, again, everyone here knows, there are large genomic databases associated with many phenotypes out in the world, of which the UK Biobank may be the most advanced, but in Finland, FinGen is looking to enroll 500,000 people. It's on its way, which is about 10% of the population. UK Biobank already has about 500,000, but we need more diverse populations, and the Million Veterans Program in the US and All of Us, which is getting going, are more focused on diversity, but diversity within America. It's going to be really important to have other global populations, again, just to not only for scientific purposes, but also because of global global health equity. But these have, along with everybody's genotypes, and now in the UK Biobank, everybody has been whole genome sequenced. Not all the sequences have been released yet, but everybody's been whole genome sequenced, and that will happen in the All of Us program in the US as well. And that will be associated with blood and urine biomarkers. In UK Biobank, about 50,000 on the way to many, many more have had brain MRI, heart MRI, abdominal MRI. There's sociodemographic data, including educational data, employment data, family income. In many of these, there are links to electronic health records, cancer registries, death registries. In Finland, we know when the patient last filled his or her prescription for an antipsychotic drug. There are surveys on lifestyle factors, and you can apply in, say, UK Biobank. If you have an interesting set of questions you want to ask and you get permission, you may get 100,000 genotyped or sequenced people responding to your questionnaire. This will allow us to generate not only clinical GWAS, right? Because once you're genotyped or sequenced, then you can run a GWAS on any phenotype based on your medical records, your response to questions, And these don't have to be medical. I mean, as you know, there's this large sociogenomics movement that has studied educational attainment and intelligence. And I don't have to tell this group that there are no shortage of ELSI concerns associated with these things. You can also send your data from 23andMe or Ancestry to third parties like Gene Plaza, and they will tell you, they will calculate your educational attainment score. You know, at some point that becomes just retrodiction, not prediction. But again, all of these scores come from publicly available data now. It's important that we, again, I said we shouldn't fetishize heritability. The heritability of a trait represents the proportion of variation in a population, not in an individual. Heritability depends on how the trait is measured. And I've just made this point about psychiatric genetics, where we're measuring still by very conventional criteria. And we have a GWAS for conventional schizophrenia, for bipolar, but actually those alleles are continuously distributed in populations. Symptoms are continuously distributed in populations. And based on our diagnostic system, not on any scientific reality, we are essentializing those, you know, as one disease or another and making diagnoses and predictions. Heritability also depends on the effective environment for the population. And again, I'll remind you of the limitations of GWAS and PRS. We can talk about these limitations for a long time, and some of these things were in some ways touched on yesterday in a way that was quite optimistic, that maybe families are more sophisticated in interpreting these things than certain famous social scientists who write books like The Bell Curve. But high heritability traits are often mistakenly taken to be immutable. They are not. There have been secular changes in adult height and IQ, and in the US, we're now getting shorter, even though the heritability putatively of height is 0.9. Our nutrition and lack of exercise and Lord knows what else is leading us actually to shrink. So there's not an endless march to progress. And we all know about the example of phenylketonuria, which is 100% heritable, but can be prevented with a phenylalanine-free diet. This misinterpretation of heritability, even if the families that were surveyed and discussed yesterday know better, has historically among policymakers produced both fatalism and damaging policies. And of course, we still have to worry about fatalism in patients. If I have a few more minutes, I think I do before questions.
Yes, you do.
Yeah. Let me just take this a little bit further to biomarkers. So in some sense, the problem with this thing that, of course, and I'm so sorry, Paul. I mean, you didn't invent it, and now you're trying to parent it. But is that there's no ground truth. It truly is, you know, committee work, even in data selection. In some ways, GWAS and sequencing and polygenic scores, maybe it's giving them too much credit to call them a ground truth, but they are connected to nature. They are derived, they are discovered, they're not invented. What we're inventing are the phenotypes that we want to name, sort of pretending to be Adam, you know, naming the animals. But they are a connection to nature, which is very positive. But they have real limitations, and we have. But in the nervous system and in much of general medicine, we can go much farther in prediction and disease stratification. And a great example is Alzheimer's disease, where in the clinical trials that were conducted 10 or 15 years ago, of course, Alzheimer's disease therapeutics has not yet yielded what people want or need, but but the original set of clinical trials focused on amyloid. It turned out that at postmortem, 30% of the patients did not have an amyloid disease. They had Lewy body dementia or something else. And in the interim, biomarkers, that is carefully studied, well-powered trials like ADNI, the Alzheimer's Disease Neuroimaging Initiative, followed people and established first in cerebrospinal fluid, and then later found which of these markers could transfer to blood, not all of them, or which could be interpreted in terms of PET scanning, some of them, that the A beta, the peptide component of amyloid, levels change well before there are any symptoms. Levels of A beta actually go down. They start, instead of up in the CSF, they start getting incorporated presumably into plaques. Another toxic protein, tau or phosphotau, starts to go up in the CSF. And now there are many, many alternative biomarkers that follow different patterns, some earlier like A beta, some later like tau. But all of these allow a therapeutic, which hopefully we will have, before I lose my keys, we'll have a therapeutic that can be entered into a clinical trial before somebody has symptoms, after which it is clearly, we know now, too late. So, I don't want to overclaim in psychiatry, but we're starting to get there. So there was a very important finding, actually from GWAS. My colleague Steve McCarroll discovered with his MD PhD student Ashwin Shekhar that schizophrenia odds ratio goes up with certain alleles of complement factor C4 that you might remember, complement proteins from the periphery that are involved in opsonizing or targeting bacteria or cancer cells or virally infected cells for destruction. In the brain, complement is at much lower concentration, but it targets weak or inappropriate synapses for being phagocytosed by either microglia or astrocytes. And frankly, I don't know which is more important at this stage. And these alleles lead to higher expression. So the higher the expression of C4a protein in schizophrenia brain, there's overlap. None of these are perfect separators, but remember, schizophrenia, what we're calling schizophrenia, is highly, highly heterogeneous. And the goal here is to, maybe these people are different from these people, maybe not. But the point is, we're starting to find proteins that have potential biological significance that are different here in schizophrenia brain, or in living people, it would have to be in cerebrospinal fluid. And so there's a large, there are actually a number of biomarker studies beginning in schizophrenia or the psychosis spectrum in CSF. And, I don't want to take you through this too much, except to say C4 and other complement proteins like C3 are eat me signals. And the idea is that people with schizophrenia have a third of the GWAS findings that we can identify, and a third of the rare variants are synaptic proteins. And maybe those synapses don't smell right to the astrocytes and the microglia, but also some people produce too much of these eat me signals. And we sort of know what these things are, and cerebrospinal fluid studies are beginning. So I showed you C4 in brain. Here's another protein that interestingly also goes down in Alzheimer's disease, that goes down in schizophrenia and correlates with neurocognitive function. These are small studies, but it's just to say that one of the reasons that, polygenic scores for predicting psychiatric illness are only the beginning. It will take decades to have good biomarkers. They're beginning with schizophrenia, but they will extend across many categories of illness, and maybe in a bottom-up way, they will help us with a better classification. I think I should stop there. I was going to talk about embryo selection based on polygenic scores, but you can tell that I'm going to say that that's a bad idea. What I think is important, though, is to remind you that besides the risk of selecting embryos based on phenotypes associated with polygenic scores that leads to disfavoring certain phenotypes, and it might be the harbinger of a new individually chosen eugenics worth of discussion. The important thing is that we only have 20,000 genes. They do lots and lots of different things. It turns out if you want to select against polygenic forms of ASDs, which is a big area of concern now in the neurodiversity movement, you're probably more likely to be selecting against your kid's IQ than against the likelihood that they have ASDs. And this is not in the ads of the 4 or 5 companies that are offering to test IVF embryos using polygenic scores. But again, this will be very rich material for further consideration in groups like this. And let me end there.
Thank you so much. So we do have plenty of time for discussion now. I see that Eric Perrins is raising his hand. Eric, do you want to unmute yourself?
Hi, Eric.
Hello, Steve. Thank you for a really masterful presentation. I wonder if you can help me think about sort of what I think of as 2 moments in your talk. There's this moment where you are, accusing the DSM of essentializing, and for good reason, of course.
Because it does.
Yeah, well, we're talking about this continuous variation,
Yeah,
which these psychiatrists go and carve up as if nature could be carved at its joints, whereas
nature is a meatloaf. That's the main point.
You beat me to that.
Yeah,
as a wise man once said, nature is not like a steer; it's like a
not a roast. Yeah.
Okay, so in the one moment we're talking about this continuity, and by the way, we're having exactly the same conversation in the context of population genetics, right?
Yeah, with race.
Right.
Yeah.
Yeah.
I think I saw.
Anna is here, and
yeah,
Anna has
beautiful.
Anna has beautifully made this point, right? Populations are meatloaf, not a roast. That's moment 1.
Yeah.
There's this other moment where you say that in the name of equity, what we really need to do is distinguish among genetic ancestries so that—
I didn't say distinguish.
Oh, Steve,
that's not the job. Yeah.
Steve, the activity of doing the kind of work you're exhorting us to do, the kind that would attend to ancestral differences, it seems to me, requires some kind of thoughtful carving up of the territory. Now, maybe I'm wrong about that, but I guess this is an invitation to recognize, for us as a field, to recognize the importance of being able to oscillate between these 2 very different ways of thinking about the problem in front of us. I mean, maybe they aren't mutually exclusive ways of thinking.
Yeah. So, I think the only reason I said distinguish is not the word we would choose. I mean, so we're doing genetic studies in Ethiopia, Kenya, Uganda, South Africa, and on the psychosis spectrum and on neurodevelopmental disorders. The former led by Carson Conan and the latter by Elise Robinson. Surely, the goal is to select people who are ancestrally diverse, but in a way that doesn't create a statistical model.
Yes, exactly.
But the output is, but we don't assume that there are that clear boundaries on those populations, right? Sure. I think that's, and it's not an ancient casuistic trick, right? Where if I think good thoughts while I'm selecting ethnic groups, it's not, it really is true that we're not looking to an output of this is not to put boundaries around ethnic groups. It's the output is really to have enough diversity, which is much greater than people think, by the way, but enough diversity in these genetic data sets so that almost anyone in the world one day who goes to the doctor and has, and if their doctor has their sequence or their genotypes, will have the benefits of genetic insights.
So I don't want to belabor the point, but all I want to suggest is that maybe when we're thinking about what psychiatrists do and what population geneticists do, maybe instead of referring to that as essentializing, I understand the respect in which it is. A more positive spin on it is they are creating categories, tentative, artificial, problematic categories that help them get on with their work today in the hopes of having better categories.
Yeah, it's true. But we yes, except for, I shouldn't keep, because there are other questions and comments, but look, essentializing is a human cognitive foible, right? It's built in, we're all grist for Kahneman and Tversky, and this is what we do. And if these were, every classification is simply a structure imposed on data to make it useful. If we really could flexibly alter those in response to data, I would agree with you entirely. I fear that something like the DSM gets reified because it's used for reimbursement by police departments, by insurance. And so it doesn't, it becomes, in the end, it defeats the idea of flexibility that you have brought forward.
Okay, so I just want to move on to another top comment from Anna Lewis.
Thanks. And I'll just quickly say something that past thing before asking something completely different. Yeah, I mean, science needs categories for sure.
Yes.
And the question is like, which are our default categories and how do we move beyond those when we realize that they have issues? Yeah, and I like Eric's invitation to think about some of the ways in which these two issues, the diagnosis in psychiatry and groups in population genetics, the analogies between them, that's a fun exercise. Yeah, I want to ask, Steve, you laid out for us kind of cutting edge of what's known as schizophrenia. You laid out for us in general your views on polygenic risk scores and what they're useful for. And I'm wondering if you can say a little bit more about where you think we are in terms of the application of polygenic risk scores, either just based on common variants or the next iteration of them, which will also include rare variants based on NGS data. Like, where are we right now in terms of their application, and where might we get to in the next like 5-10 years?
So I think we, given all the limitations, we ought not to think of using these in the clinic. However, if you were doing a human study, a clinical trial, an imaging study, on the one hand, this could be a focus of the study, right? In which case you would use polygenic scores, perhaps as a dependent variable. But I do think that they have a potential use more diversely in human studies, ex post facto, right? Meaning as you're not to overinterpret or misinterpret your results, but to understand your results as you're planning your next experiment, because they may give you, they might not, but they may give you a hint as to how heterogeneous your population was. And if people who are really loaded, if people in your depression study are really loaded for schizophrenia risk, even though they don't have a whiff of schizophrenia, that would suggest they're more severe and maybe different from some of the other people. Type 1 and type 2 bipolar, type 1 is more like schizophrenia, type 2 is more like depression. These are not categories but there are quantitative differences that are clear. That could help you in understanding your findings, but I don't think we're near either selecting patients for studies by polygenic score, especially given the Eurocentrism of the work that has preceded it. But also, but I do think it is useful in research, ex post facto, and not in the clinic.
Just a really quick follow up on that. I mean,
Yeah.
if the main use case is that ex post facto understanding the heterogeneity,
Right now, yeah.
the genetic heterogeneity of your population, might that not suggest that we should directly try and build tools which help us understand that heterogeneity, rather than, which might be different than these different polygenic risk?
Yeah, I think that would be extremely useful. I would add one other use case that the sociogenomics researchers use, which has to do with stratification. What they say is, look, if you're studying school performance, you might stratify by parental income or by school quality, because you could otherwise really fool yourself about the utility of your intervention. And why not also stratify or use as control polygenic scores for educational attainment or other things? You know, I think that's really fraught, but I think in fairness, the people who perform those studies bring that forward as one of the ethical and meaningful uses today.
We did have a comment in the chat. Betty, are you here? Because I'm not quite sure that I understand your comment. This is from Betty Levin. I'll read what she wrote, but
I am here.
okay, great.
Right, and I really know very little about much of what you're talking about, but as an anthropologist, I know that it's problematic to talk about genetics on a continental level. So if you're talking about genetic variation, there's a huge amount of genetic variation in Africa. So,
sure. Yeah.
you know, it would be really good if you can look at the associations with various genetic clusters.
Yeah, absolutely.
You know, and really get away from the continental terms, other than to say, yes, we have to study people in varied areas.
I agree with you 100%. So the cohort in Ethiopia is more different than some cohorts right across the border in Kenya than any two Europeans, right? And yes, that has to be taken into account, but not by us. It has to be taken into account by the investigators in sub-Saharan Africa, number one. Number two, the environment. So comparing the actions of genes in different environments, of course, is incredibly valuable. Most of our African colleagues still think it's too hard. The environment is an infinite search space. And we have our priors as to what matters. For schizophrenia, urbanicity, migration, I mean, get serious. Those are proxies.
They are huge, right? Yeah. Environmental is even.
Yeah, but the hope of doing this will be to be able to dissect some of the environmental risks, undoubtedly. But we're just
even for the genetic risks, it seems it would be helpful to the way people conceptualize race and genetic differences to try to get away from referring to the large geographic areas on a continental level. I mean, even within Europe, we know there are different populations.
Yeah.
Everybody here, Betty, I think agrees with that. I was trying to point out that Steve's endorsement of Alicia Martin's argument assumes that Europe is a continent with there's an ancestry that's similar enough so you can do a useful analysis.
Although we all know better, right?
We know better, and I think we, not this group particularly, but we need to be reminded that there are different subpopulations
Absolutely.
that are way
Absolutely.
different everywhere, and especially
Absolutely.
in Africa.
And
Absolutely.
my only
no,
disagreement with
prior-
my only disagreement with Eric, it wasn't really a disagreement, was the way these things get ossified. And we have to be really careful
Oh, yeah.
that what should be rough and ready categorizations for the purpose of this stage of scientific analysis doesn't take on a kind of reality, as has happened with continental differences and as has happened with DSM diagnoses.
Yeah,
Okay,
absolutely.
no, it's,
And I think that
I'm learning
going-
a lot, a huge amount from this conference. Thank you.
great to hear, Betty. And, this is a good segue because our next speaker actually comes to us from South Africa.